Yeah, so our laboratory is completely designed to discover new drugs against new targets. So we were all set up when COVID actually happened, and we really the first drug that we came across that looked like a real contender, we actually identified back in Easter. And we've been working on it ever since. So we're now at the point where we have manufactured a drug for human trials. We could be in humans as early as 4 to 8 weeks from now. And our drug is, certainly in the laboratory, it's a lot more potent than anything else currently being tested on the market. So we're not looking at a vaccine. No, vaccines are great, but take a long time to develop. We're actually looking at a drug that should, after one dose, kill the virus or stop it being able to replicate and let your body remove it from your system. So yeah, very exciting things. So we get the basic data out of the lab on that the drug targets the target. We then contracted to a Melbourne business, so it was independent of our lab, that they can test in an independent study. So that gives people more confidence, and they did the live virus work with the virus and using human cells, and they reported back that the drug was very effective against the virus and stopped it being able to replicate in those human cells. So the next stage you have to do is you write a clinical trial. You then submit it to what we call an ethics board, who review the trial, and then you're then able to, if they give the green light, you're able to go straight into patients then. So going into patients requires you to kind of negotiate with the hospital that you want to go into, or the health authority that you want to go into. But those processes can be relatively quick if the drug looks like it's going to give real benefit to patients. Yeah, so it is a really rapid development program that we've done. So really, I came up with the idea on the weekend before Easter, and then we spent all week, and even into Easter Sunday, we worked to try and determine whether this drug was actually going to work, that we identified, and then we found out on Easter Sunday that, yeah, indeed it was working. And we've basically been doing a lot of non-animal studies on cells, human cells, and we find our drug is extremely potent, as in 1,000 times more potent than the drug Donald Trump has been taking, which is quite exciting. And we're about 15 times more potent than remdesivir, which is the #1 drug that people are using just now for coronavirus. So we're pretty excited about it. We think a single dose will be enough to incapacitate the virus and move on. So we're hopefully we'll be in patients within at least 8 weeks from now. And it looks like it's still continuing to get worse at this time. So we still we're pushing hard to get this done. So developing a drug is not cheap. We've managed to really develop it to this point using philanthropic money and money from one of our commercial partners. We're moving to the next stage so we've manufactured some drug ready for our trials, and that's really expensive. We've got enough drug to maybe do about 400 people in total in the trial, and then we have to remanufacture and start again. So manufacturing a drug costs about $1 million. To make for the clinical trial, and then the clinical trials themselves are maybe about $20,000 per patient it will cost us, and we'll use maybe in the first stage of the trial maybe 20 patients here in Australia to start off with, but we want to get the drug out as soon as we can to the US and to Europe and South America, etc. Yeah, the government has a number of schemes. We've actually applied to one of those schemes just now, the Queensland government. We're also in talks with the federal government about providing the funding which will allow us to go into patients in 8 weeks' time. Currently, we don't have the funds to do that. So we need external funding. So we're currently in negotiation with both the Queensland and the federal governments about an investment into the product so we can get it and test it sooner rather than later. Because as soon as we test it, we either know if it's going to work really well or it's not going to work as well as things out there just now. But we really need to know these things quickly in the current environment.